Our next speaker is Dr. Sayoni. She's a computational biologist and leads the research and development of bioinformatics pipelines that generate biological insights and support drug discovery programs at PrecisionLife. PrecisionLife is a leading computational biological company driving precision medicine in chronic complex illnesses. You might have heard from them because they have been partnering in the programs like DecodeME, long COVID-ME stratification, and DecodeME on phenotyping. They've also been launching a big cooperation with Metrodora Institute in the US for clinical trials. And I think stratification and finding targeted drugs is exactly what we need. I'm so proud that Dr. Sayoni is now here to tell us more about how this sheds more understanding in those two conditions. Thank you so much. Dr. Das, are you ready?
Yes, thank you, Diana.
Yes, perfect. Thank you so much. The stage is yours. I'm going to mute myself now. Thank you.
Thank you. Thanks a lot, and thank you to the UniteToFight team for the opportunity to speak today. I'll be talking about PrecisionLife's unique approach and how we are using it to generate new insights for ME/CFS and long COVID. So thank you so much for giving the opportunity to speak to this in this platform today. So at PrecisionLife, we are reimagining the ways to analyze patient populations in order to understand what are the different drivers of disease biology and how do they relate to different patient subgroups within complex chronic diseases. PrecisionLife has developed a unique analytics platform to do this, and our starting point is always patient data. And we particularly like data sets that has information on genetic data and longitudinal clinical history and other types of information, because the platform can analyze all of these different types of data and bring them together to generate insights about a particular disease population. We have over 30 data access agreements with patient charities, research consortia, and other organizations where we get access to a variety of data sets. And what we're trying to do with these data sets is shown by the graphic in the middle. So this shows a mechanistic patient stratification of an Alzheimer's disease patient population. All of these patients have the same diagnosis of Alzheimer's. What we've been able to do is substratify them into different subgroups. So the colors here represent a set of SNPs or single nucleotide polymorphisms that are shared by different patient subgroups. Each of these groups may have different clinical presentations, such as different sets of symptoms or severity, and will perhaps benefit from a different type of medication that is specific to their disease driver. One of the most useful things about this analysis is that we can tie these results back to individual patients, and that allows us to look at prevalence of particular mechanisms and also look at potential repurposing opportunities if available. So for example, here we find 6 major subgroups of Alzheimer's, and we can see that lipoprotein metabolism is the most prevalent one. And this is where a lot of the drug development has focused so far in Alzheimer's. But we see only that relevant to 1/3 of the patients. And this goes a long way to explain why it's been so difficult to put some of those therapies into the clinic without any additional level of stratification insights that would help us point the mechanism back to particular sets of patients who are most likely to benefit from it. So what enables us to generate such detailed insights? In large part, it's because of the way we go about analyzing chronic disease data sets. It's different from the traditional genomics approaches, such as genome-wide association studies or GWAS, that are looking for single variants in genes to explain disease. So we know that chronic diseases are multifactorial and has many causes and effects and can vary enormously based on combinations of different features. So in our approach at PrecisionLife, we are actually looking for these combinations of features using a hypothesis-free, data-driven approach that is not based on any prior knowledge. And we analyze how these interactions from multiple relatively common factors come together to trigger the disease processes. For example, here is a real example of a 6-SNP disease signature that we identified in 150 women with the pathogenic BRCA2 variant which was found in a set of patients who had not developed breast cancer by the age of 55. And what we can start doing from this disease signature is to formulate a mechanism of action hypothesis. And what we think is happening here is that there's a partial blockade in the insulin receptor that's a key activator of the oncogenes PI3K. And if the oncogenesis is not activated, it prevents these patients with this disease signature from developing breast cancer. So such an insight can have potential utility in the clinic, but it also allows us to start stratifying patients who have particular underlying disease mechanisms and generate patient stratification insights and more personalized risk scores as well. And I'll be talking through all of these analysis that we have done in particularly ME/CFS and long COVID. So we have so far analyzed over 40 chronic disease datasets through our combinatorial analytics platform, but particularly will focus the rest of my talk on the ME/CFS and long COVID analysis that we have done. So at the time of our study, there were no known reproducible genetic associations or any known common pathophysiology that was ascertained from just purely genetic analysis. Using combinatorial analysis, PrecisionLife had identified genetic insights into the mechanisms underlying both ME/CFS and long COVID, and I'll share those insights in the next few slides. We aim to validate and extend these insights in a wider group of patients from the DecodeME study as part of the DecodeME project that I'll also talk about after that as well. So for our ME/CFS study, we analyzed 2 populations from UK Biobank. There were 2 sources of data for us, and we used for our primary analysis a dataset made of individuals who had reported diagnosis of ME/CFS in UK Biobank's pain questionnaire. And we also formed a secondary dataset of a disjoint population who had self-reported chronic fatigue syndrome at the time of recruitment in a verbal interview. So from the primary pain questionnaire dataset, PrecisionLife platform identified 84 combinations of around 200 SNPs that are strongly associated with ME/CFS risk. This analysis identified 14 genes that we were able to look into in more details and map them into 4 main mechanisms that related to ME/CFS. These were viral or bacterial susceptibility, development of autoimmune response, metabolic dysfunction, and sleep disturbance. So all of that analysis was based entirely on the data that was generated from a hypothesis-free analysis, and we started investigating which of these genes were enriched in the patient subgroups that we identified. So this particular figure shows the mechanistic stratification of ME/CFS from the pain questionnaire dataset. Each dot again represents a particular SNP or a single nucleotide polymorphism identified in the disease signatures. We then clustered them together based on the patients in which they were found, and the colors represent the resulting patient subgroups. So when we start looking into some of these subgroups, if we see the green patient subgroup here, we mapped that to the gene ACAP1, which could be linked to fatigue presentation due to defects in mitochondrial respiration associated with AMPK. So this is likely to be involved in the post-exercise recovery and would explain a lot of the impaired recovery and severe fatigue symptoms that we've been hearing about. This is potentially one of the underlying mechanisms of a subset of patients who would have this phenotype. When we looked into the clinical data available for these patients, we saw a higher prevalence of fatigue phenotype as well. In contrast, we found another patient subgroup, which is shown in orange here, which was mapped to the clock gene, which is one of the key regulators of circadian rhythm. An alteration of circadian rhythm is hypothesized to contribute to many of the symptoms experienced as well, including insomnia. So this really starts illustrating how by focusing on particular patient subgroups, we can develop mechanism of action hypothesis and then start to understand which targets and drugs are likely to better work for certain groups of patients. We are not disease experts at all, but we were able to look into these patient subgroups and mechanisms just by following what the data was telling us from a hypothesis restudy. So the insights from these studies was supported by the replication and cohort comparison analysis we did with the secondary data set. And we are currently replicating these insights in DecodeME's much larger, better characterized populations as part of the LeuCOME grant that is supported by Innovate UK. And for the long COVID study, we generated two populations from the Sanogenetics long COVID goal study. Majority of the participants in this particular study were likely to be affected by the wild-type SARS-CoV-2 variant when we looked at when they were diagnosed with COVID. And we had information about the genotypic data and long COVID symptoms. So we used the available information for this cohort to generate two populations that we used for our analysis. We created a severe cohort where cases reported the greatest and the most severe symptoms across many different areas. And we also created a fatigued dominant cohort where the cases predominantly reported fatigue-associated symptoms. We then analyzed it through our PrecisionLife platform and then identified 73 genes that were highly associated with at least one of the long COVID populations. We saw some similarities between the two cohorts, but we also saw certain differences between the two cohorts. And one of the most notable differences was the fatigue cohort had more similarities with some of the genes that we had identified in our previous ME-CFS study. We also noted that there were 14 genes that were previously reported in a differentially expression analysis in a previous publication reported using long COVID data. And we also looked at some of these genes that were associated with acute COVID infection. And these were generally more enriched in the severe cohort. However, we didn't have enough data to ascertain any of that in this particular cohort. And we are hoping to get access to larger long COVID data sets, which are better characterized so that we can dig into some of these questions a little bit more. But one thing that I mentioned already, we saw higher overlap of the ME-CFS genes with the fatigue dominant cohort of long COVID. And two of the genes wanted to mention were ACAP1 and clock that I had just shared from our ME-CFS findings. So building on from the results of our previous analysis, we are now extending all of those analyses into the LeuCOME project. So this project will extend PrecisionLife's previous insights to include deeper multimodal data that is now available from the DecodeME study, which is the world's largest study of ME-CFS with over 18,000 participants, led by the DecodeME partnership, which includes Action for ME and MRC Human Genetics Unit at the University of Edinburgh. We aim to replicate the disease signatures we identified before in independent samples in DecodeME, and we aim to use the insights generated from this project towards development of tools for triaging patients and identify potential repurposing opportunities. The LeuCOME project is an early adopter of the new patient and public environment standard for medical research known as PPI, and it brings a different perspective, the lived perspective to research, and this can help ensure highest research standards that enable researchers to actually focus on the questions that patients consider to be most important. So PrecisionLife is currently working very closely with the LeukoMe team on the first 2 batches of data and seeks to capture the invaluable insights from our PPI partners in relation to the particular study design, interpretation of questionnaire data, and analytical results. So I'll dig into a little bit more of the genetic triage tools that I just mentioned. So the UK Biobank ME-CFS study generated these 84 combinations or disease signatures for us. When we started looking at counts, just simply counting the disease risk signatures in cases and controls, it illustrated the predictive power in differentiating in cases and controls. So it starts to pave the way of reducing these insights to practice. Following further application studies, we aim to develop these into genetic triage tools that would test for these disease signatures in patient data and help clinicians to triage patients presenting with disease symptoms, and most importantly, also enable them to point those patients back to potential underlying mechanisms that might be underlying their form of the disease. This enables us to start thinking about clinical trial design and how we might go about recruiting patients who are most likely to benefit from targeting a particular mechanism. So we refer to this new generation of genotyping triage tools as triagnostics, and it aims to provide a lifetime risk of developing ME/CFS or long COVID, along with potential mechanisms driving disease where possible. So PrecisionLife has an active collaboration with the Metrodora Institute in the US, which is a research center focusing on neuroimmune access disorders, to develop these triagnostic tools. Following further analysis as part of the LeukoMe project, the partnership aims to develop the first tools in long COVID and ME/CFS. The partnership has announced recruitment up to 1,000 patients starting from next month to evaluate the clinical predictivity of these tests that aim to provide clinicians with an informed view of a patient's risk of a specific indication and which mechanisms a particular patient might be exhibiting. So these tests would be intended to be employed in clinical settings alongside other existing clinical criteria so that it can aid diagnosis of these patients with potential symptoms that are consistent with ME/CFS and long COVID and help the clinicians to aid early referral to probably specialist clinicians. Ultimately, we aim to combine all of those insights in ME/CFS, long COVID, such as the risk scores, the mechanistic stratification, and the potential repurposing candidates for each of the prevalent mechanisms in the 2 indications, where there are well-tolerated medicines that are gone beyond phase 3 or generics are available for these mechanisms. We plan to use those genotype tests to start recruiting smaller cohorts of patients with particular mechanisms, and we then aim to evaluate the effectiveness of any of those repurposing candidates that target their mechanism. So we are actively working with our partners on these projects. However, all of this is subject to testing at various stages and which takes time. So we regularly share all of our updates on our studies in ME/CFS and long COVID projects, and you can use this QR code or this link in our website to follow those updates. And we also are part of certain events in collaboration with our partners like Action for ME, University of Edinburgh, where we host or organize events like this biohackathon, where we engaged early career researchers to use the data that we had generated and see if that can be used to pave the way to answer some of the research priority questions set by the ME/CFS Priority Setting Partnership. Finally, I would like to thank all the study participants who have provided their data to make this research possible. And also thanks to the DecodeME team, including Chris and Sonia, the DecodeME team, the Metrodora Institute, and also the entire PrecisionLife team for their contributions towards these projects. With that, I wanted to thank you everybody for listening in today, and happy to take questions.
Thank you very much for sharing data, and I am sure that this approach will lead to a speed up in finding solutions, treatments, and identify people at risk to developing ME or long COVID in the future. So thank you so much. I'm going to turn to the questions from the audience. So the first question is from Tim. He's asking, isn't it crucial for research to differentiate between long COVID and COVID without post-exertional malaise and not label them the same?
We would love to. The thing is, we are sometimes, most of the times, constrained by the data available to us. So we have, in our long COVID data set, we didn't have detailed information about symptoms at all. So what we used was a symptom rating scale that we had received from the participants who had said, how do they feel before and after long COVID, and used that to assess whether certain patients had higher severity of fatigue phenotype versus patients who were reporting more breathlessness or other sets of symptoms. We would love to work with data sets where there's more deeper characterization, but I'm also aware of the constraints that we have in getting a right diagnosis and having all of the symptoms properly encoded like we would want. The DecodeME study particularly has really great questions that we are hoping to utilize in order to dig deeper into the ME-CFS analysis. We are really looking forward to do the same in long COVID, and would appreciate if there's any data sets that you have in mind, please let us know of it. I'll be delighted to include that.
Thank you so much. That was an outreach to the community. I think we have over 1,300 listeners. I think someone maybe come up to you and suggest that.
We would love that. Thank you.
Thank you. We are here to unite to fight. So let's work together. Perfect. So I have another question. Are individual precision medicine analysis already available today to long COVID and ME/CFS patients? Is there anything on the market yet?
As far as I know, I'm not aware of any such tools, but I am aware there are particular research projects going on in independent clinics, probably put together by clinicians. But I'm not aware of any product at this point in the clinic.
Okay, thank you so much. So I'm turning to the next question. Does identification of significant genes get us closer to a treatment cure, and how?
So the significant genes point us towards genes, particularly mechanisms that we think, if they have a sensible biological mechanism of hypothesis tying from all of the biological knowledge that we've got, that allows us to investigate, are there potential disease modifying therapies or drug candidates out there that could be used to recruit patients with that potential disease mechanism and investigate whether potential off-license usage or other disease modifying therapies would be useful. And it need not be, or they might also benefit from certain symptomatic reliefs from those mechanism of action hypothesis as well. So it is a way towards getting into the mechanism, which might not be very easy to investigate from such a heterogeneous patient population.
Okay, thank you very much. That's very interesting to know. Perfect. So We have another question here. Are you also looking at vessel inflammation? Is anything pointing to that?
We've seen a number of genes and pathways associated with inflammation, so chronic inflammation, neuroinflammation, but and we have seen overlapping patient subgroups who have aspects of various aspects of that. So yes, I think there are aspects of it, but eventually we will be sharing more when we are confident of those results in our wider population, such as DecodeME.
Perfect. Thank you so much. So let me see what else in question we have. Yes, someone is asking, have you tested for special SNPs like SMN1 and SMN2 in muscular atrophy? Is anything ring a bell in that context for ME/CFS patients?
Not that I recall. We did not have detailed information. When we started looking at comorbidities or known literature, we could not see that. But I have made a note of it, and I will look into that if we've anything. We've not come across those genes in our patient population, but also it's important to keep in mind the UK Biobank study that we have done represents only a small subset and may not capture all of the heterogeneity that we are aware of in the entire patient community. But we're hoping to incorporate that in.
Perfect. Sorry, yeah, so we can also send you over the questions if we can't. I think we were not able to have all the questions read out to you, but we can send them over to your way.
Yes, I'll be happy to respond in any way that was convenient.
Perfect. So let me I hope I didn't skip an important question, but I'm seeing here, what are the options for the very severe patients based in other countries in terms of accessing some of the testing that you do?
We just starting that in the US with the help of Metrodora Institute. I think any insights that come out of that would be beneficial for the entire patient community, and we'll be sharing those insights, and it should have a positive impact in helping access to those kind of tools in future, irrespective of countries. But that's something that we will definitely share widely when we have information. And just a note on the severity of the patients, that's something that we had inferred for the long COVID cohort. We had no information about severity from the UK Biobank ME/CFS population, but we do have that information for the DecodeME cohort, and thanks to the DecodeME team for including all of that characterization. And we are hoping to use that to generate more novel insights and improve this.
Perfect. So maybe one last question, but I think you're still in the clinical trial phase, but someone is asking, according to your data, what are the practical implications for treatments? Is there something you can say so far?
So we're just starting that, so we will just start recruitment, and the clinical trials will start after we've finished replication studies in the Leucome project. So this, as I'd mentioned, these tools would be employed only in a clinical setting and are meant to guide clinicians who are facing patients basically who have overlapping symptoms or symptoms consistent with ME/CFS and long COVID. We know that the criteria is not widely accepted, so it is going to enable them to understand whether these disease signatures point towards a particular indication and also point towards certain mechanisms specifically and help aid that process. So it is a lifetime risk, high risk. If one gets high risk, obviously there are other external factors that we are aware of, could be viral, non-viral triggers. Unfortunately, this is a genotypic triage tool, so there are going to be, we will empower the clinicians to make that decision and help that.
Thank you very, very much. The time has unfortunately came to an end, but I'm really happy that we had the chance that you could present your data, and we're very delighted that you made the time to come here today and talk about your project. So thank you so much, Dr. Das.